Have you identified the ideal candidate to push into the clinic? And are you wondering how to select efficacious doses for first-in-human clinical studies? It is an essential step for any medicine. Do start the thinking process early. In this video, I'll show you strategies to set target efficacious concentrations for first-in-human clinical trials. The first point is that the strategy depends on the mechanism of action. And so, we'll see first how to set the target concentration for an inhibitor based on in vitro potency, then for an inhibitor based on in vivo data, then moving to agonists, first based again on in vitro potency, and then on in vivo data. A disclaimer, this video is not about modeling human PK from animal PK. I'm talking here from the pharmacologist side, and I truly advise you to hire a PK modeler for the second part. So, first considering inhibitors, starting from in vitro potency. Let's assume you've characterized the potency of your inhibitor in an in vitro functional assay. The target here is to reach maximal inhibition, so 100% inhibition, and to do that at the lowest concentration possible. A proxy for this concentration is to use either 3 times the IC90 or 10 times the IC50 on a 4PL dose response curve. You can appreciate from the picture here that both values land more or less on the same concentration and are very close to 100% inhibition. You can apply the same principle if you have characterized in vitro receptor occupancy instead of using a functional assay. Important points if you are using this strategy is that you need to consider the free concentration or the protein adjusted concentration. You also need to make sure that the in vitro assay used or the percentage of receptor occupancy are representative of clinical efficacy. If you can, use prior clinical knowledge to verify the validity of this assumption. For instance, with an inhibitor of the same mechanism of action but different chemical class. Another essential point is that you need to consider the concentration in the human target tissues. So often in a clinic, you will measure concentrations in plasma. However, the inhibitor needs to be active in the target tissues. For small molecules, you can use biodistribution studies to evaluate tissue to plasma ratios in humans. Alternatively, if you are using free concentrations, the Kpuu is often close to 1 in most tissues. You still need to have a good knowledge whether the inhibitor is a PGP substrate or the substrate of any other transporter. For large molecules, you can similarly use biodistribution studies if you have them. And alternatively, to give you a first idea at least, Shah et al. In 2013 have evaluated ratios in different species for approved monoclonal antibodies. You can see the picture below for the different tissues. Another essential point is that the concentration needs to be maintained at all times. Indeed, you want to reach 100% inhibition at all times. So the target concentration should represent the C mean or sometimes the C average. Let's now move to the strategy for an inhibitor, starting from in vivo efficacy data. Here, the goal is to target the plasma concentration where full inhibition is reached. Here, you will need to build a PK-PD relationship and, most importantly, to consider an in vivo model and outcome which is representative of human clinical efficacy. Once more, you want to reach 100% inhibition at all times, so it is important to consider the concentrations measured in this model as the C mean or the C average or sometimes the AUC. You can also evaluate in the same efficacy model which one is the most appropriate, meaning which one is driving the efficacy. Now moving to agonists, starting from the in vitro potency. You can start from the in vitro concentrations yielding the desired receptor occupancy. So you will need to have a good knowledge of the pharmacology and the mechanism of action of the agonist to know how much receptor occupancy is necessary, so which percentage is needed. Is it 10%, so very little receptor occupancy is enough, or closer to 30 or 50%? You will have to build that knowledge upfront. Be aware that for some mechanism of action, for instance ADC or ADCC, the effect is independent of the receptor occupancy. Another point that is not built here in vitro in receptor occupancy assays is that targets have inherent turnover rates, which can be changed upon agonist binding. So it will be essential to evaluate this in another type of assay. Sometimes K-on and especially K-off can be more meaningful than KD or pure receptor occupancy. This can be particularly true for monoclonal antibodies. Also for mAbs, concentration in receptor occupancy assays can come close to the concentration of the actual receptor. And if that is the case, you will need to adapt modeling, taking into consideration the law of mass action. You may also start from in vitro functional assays. You may want to reach an EC50 here, but if you already know that you actually need very little activation of the receptor to have the desired clinical effect, you may use the lowest effective concentration, or LEC, or the minimal effector concentration, MEC. This concentration is defined as the concentration where the dose response curve crosses the following threshold of the mean or median of the cell control plus 2 or 3 times the standard deviation of the cell control. This is illustrated here on the graph on the left. As for receptor occupancy, it is essential to have a good knowledge of the relationship between the in vitro assay and the desired clinical effect. Two attention points here. The first one is that the target concentration may only be reached at Cmax for an agonist, so it may not always be necessary that the concentration in the target tissue is higher than the target concentration. In other words, the target concentration only needs to be reached at Cmax. And second point, similar to antagonists or inhibitors, it is essential to take into account expected biodistribution to target tissues. So once more, you will measure the concentration in the clinic in plasma. However, the agonist needs to be active in the target tissue. Moving to agonists, but starting this time from in vivo data. Here, similar to inhibitors, you will need to build a PK/PD relationship. If you only need a slight activation of the receptor or target you're considering, you may want to use the MABEL approach. MABEL stands for Minimum Anticipated Biological Effect Level. This is illustrated on the graph below. You can see the blue line representing the efficacious effect and the red or orange line representing the tox effect. Instead of looking at nowhere else, you may want to consider first the blue line, so the therapeutic effect, and start with the lowest concentration, which gives you just a little bit of efficacy. This would be your very first dose or concentration to test in humans. Attention points for this strategy are the following. Similarly, from when you're starting from in vitro data, the target concentration that you need to consider may only be the Cmax. You will need to establish and verify this, but this might be the case. As you are using animal data, it is crucial to consider interspecies differences in binding to the target, age of the animals, which may be very different between an animal and a human, differences in pathway as well. For steep dose response curves, it is also crucial to consider higher safety margins. For instance, for TLR agonists, you often have a kind of on-off response. So here it is really advised to start either from the LEC or from the MABEL approach. You may also consider a split dose approach to dosing. For instance, you only give 10% of the dose on day 1, 30% on day 2, 60% on day 3. All right, here we are at the conclusion. So in summary, it is essential to know your pharmacology. Do start early thinking about the strategy for first-in-human dose selection. You may want to combine multiple approaches to setting the target concentration. So aligning in vitro and in vivo data. You may want to build a buffer for inter-individual variability in PD responses. You will, by the way, do the same for PK responses. Setting target concentration is a difficult exercise, and you will not be entirely sure that this is the right concentration before you've actually moved into the clinic. So for this, it is important to be clear and transparent about the underlying assumptions in the quality of the data you're using, the risks, and the assumption you've made. To handle uncertainty, you can perform sensitivity analysis, worst case scenarios, best case scenarios, and robust design. And finally, it will be essential to monitor properly during clinical studies. Finally, the next step to identify the first in human doses is to predict human PK. As mentioned at the beginning, for this, I truly advise you to consult a PK modeler. I hope this video was useful. And you, do you have any advice on this very interesting topic? Be sure to leave a comment. Bye bye.